Hey everyone, welcome back. Today we're talking about something that we have done a few times already and you might be going through as well, which is interviews. And let me tell you, they're always unique because they want you to know biology, maybe computer science, probably some statistics, and probably the hardest part is explaining everything clearly to the interviewer. So,
Exactly. So let's break down what actually matters when you are interviewing for junior to intermediate bioinformatics positions. Today, we are going to cover mindset, technical prep, and how to communicate effectively, because that last part is where most people struggle.
yeah, so let's talk about mindset first. I think a lot of people think that they have to memorize every algorithm, every tool that's out there, every parameter, but that's not actually what they're looking for. They want to learn in this interview process how you think. Are you able to break down a problem into smaller chunks, or could you explain the reasoning for why you're doing what you're doing? Ultimately, they want to know, can you connect the biology to the computation?
Yes, so it's less recite an algorithm from memory or from a book, and more explain when you'll use global versus local alignment and why. That will show you problem solving process, not just what a definition of an algorithm. So learn to structure your answers using the STAR format. I should have learned this sooner in my life. It's a stand for situation, task, action, and results because it helps you all to tell a clear and compelling story about your work.
Right, I agree. I think STAR is super useful, and we'll come back to it. But I wanted to mention something practical as well. You want to find out early on, for example, in the initial call with HR or the recruiter, what the interview process is going to be like. You need to know, are you going to prepare for a whiteboarding exercise? Should you prepare for a presentation? Or is there going to be a live coding session in your near future? You need to know this early so you can prepare. So just remember, one of the first questions you should ask HR or the recruiter is, what is your interview process and what's the timeline?
Yes, don't assume every company or every place is different. Now, let's talk about what they are actually looking for more specifically. There is the technical stuff, obviously. I mean, we are in bioinformatics, but it's not
Right?
just can you code. We assume that almost everybody will be able to do that. It's more like can you translate between worlds? Can you explain biological concept to a software engineer? Can you explain a computational method to a biologist? That's a real skill we are looking for, or hiring managers are looking for.
Yeah, for sure. And another thing they're looking for is, can you handle messy data? Because real world data is super messy, especially in biology. So, for example, do you know when your results don't make biological sense? Like, let's say your code runs fine, it spits out some numbers or some results, but the biology is wrong. Are you able to spot that? And I've seen people get really focused on making the pipeline work, but they forget to ask, wait, does this actually make sense from a biological point of view? That is something you definitely want to nail.
Yes, I cannot say more than that. That is huge. We want to see that you can think critically about your results, not just able to produce them, or even if they are like produce nice reports, it's like what what they mean. And honestly, we want to know that you can communicate clearly with people who have different backgrounds, as we mentioned, because informatics is all about bridging gap between different disciplines, like many different ones. It will depend on the role.
So let's get practical about what you want to prepare. Your core toolkit is probably going to be Python or R. So you want to be really solid with data structures, loops, functions, and how to manipulate data sets efficiently. Command line, I think, is non-negotiable. You want to know how to use grep. If you can use awk or sed, I think that's awesome. It can really make your life easier. And then you really want to know Git basics as well, because if you are in a professional setting, you need to know how to do version control.
Yes, and then know at least one tool. Like if you're in sequencing technology, probably an alignment tool is a good example. One that is well standard that you have used, so BWA, STAR, and you really need to know very well what is the alignment doing. Every alignment has a different way to approach the problem, and it's good to know the limitations and the application of each of them. And as well, statistics. I mean, statistics is critical. Understanding what p-value means, multiple density correction, basic distributions, when to apply one or the other. People sometimes don't pay that much attention to that, but you use it all the time. So you need to at least conceptually understand what is going on there.
Agreed. And then file formats matter too. You want to know the difference between a FASTQ file and a BAM file. What's a VCF file versus a GTF file? You want to know what's in these files and when to use what. And if you don't, that's going to be a red flag to the interviewer. The same thing with sequencing technologies. You want to know the difference between the big three, which is Illumina, PacBio, and Oxford Nanopore. And here's a typical question they might ask: walk me through an RNA-seq pipeline. So you want to have a solid answer prepared. You want to make sure you cover quality control, alignment, quantification, differential expression, and interpretation. And the cherry on top is, you're going to say, there's a QC step. I can do it every step in this pipeline.
Now we are going to switch gears a little bit. Let's talk about how you communicate your answers, because this is where candidates really succeed or struggle many times. So when they ask a technical question, don't just name drop tools. Explain why those tools. It's better to just say 2 tools and why than like 10 and leave it at that. So a bad answer is, I will use this SIG2 because it's the standard. A better answer to that is, I will use the SIG2 for differential expression because it handles log counts data well through its negative binomial model, and that is the best distribution that can explain sequencing data.
Yeah, so do you see the difference? You want to show that you understand the why, not just the what. And this is especially important when you're doing like a whiteboard or screen share problem. You want to restate the problem first, make sure that you understand what they're asking, and then you can ask clarifying questions. What's the organism you're looking at? What's the sample size? Is there any different type of data that's involved in this problem? And then you want to outline your approach before you start coding or drawing. You want to think out loud because silence sometimes makes interviewers nervous. So keep filling that silence.
And it will happen, that is normal, that you won't know the answer all the time. And you can be honest. I don't know all the time the answer. So be honest and say, I haven't used that tool or I haven't had the opportunity to really address that problem. But I will approach it this way. So I need to look more into maybe the exact parameters, but this is the general workflow that I will do. But never make up answers about methods, tools, or anything, because it's very easy to catch up and that will destroy your credibility.
Right, that's good advice. Be honest, don't make it up if you don't know. It's okay. So let's talk about how you want to present the projects you've worked on in the past. In my opinion, project discussions are huge. You want to pick the best project that you think you worked on for a given challenge, and you want to know it inside out. So be ready to discuss the biological question. What were you actually trying to answer? Not just saying, hey, I did RNA-seq on this, but you want to phrase it like, so we wanted to understand how X affects Y, and this is why I picked this problem solving approach that I did. And you want to share details like, organism or sample size or the quality issues that encountered. And I think it's super important to have at least one project that you worked on in R and a different one in Python, because the question might be, hey, walk me through how you provided a solution in Python. And you won't be able to answer that question if you only prepared your R problems.
Absolutely. And be ready to discuss challenges. What went wrong? How did you fix it? What would you do differently next time if you could repeat it? This is where the star format really shine. So set up the situation, explain the task you faced, describe your actions, and share the results. Don't just say, we found 500 differentially expressed genes, yoo-hoo. Explain what that tells us about the biology or how we can approach to make sense of these genes.
Yeah, and here's a red flag to avoid. You don't just want to say, I ran the default pipeline. That shows no depth of understanding. Confusing correlation with causation is a big one, ignoring batch effects, or not validating unexpected results. These are all bad signs. Instead, you want to show the interviewer that you can go deeper. You can show them that you know, for example, GC content bias in your samples. How did you apply a correction? What kind of correction did you choose for this problem? Or maybe after seeing the PCA, I realized we had a batch effect and it rested by controlling for batch in the model. You want to show the interviewer that you can go beyond the surface.
Okay, now Let's talk about how to really stand out, if possible. I mean, there are many good people around, right? So, my top advice will be prepare thoughtful questions that show you are thinking about the role that you are applying strategically. Ask things like, what does a typical project timeline look like there? Or how do computational and wet lab teams collaborate? This question actually demonstrates that you are thinking about how you will work there, not that you are, if you get hired or the project only.
Yeah, how do you want to stand out? I have two top ones. One is I would recommend you ask a question about how the team works together from every interviewer you meet, because that gives you a better rounded picture of the team you end up in, right? You want to have a baseline that you can compare to. And the other thing I would recommend is read some recent papers from the lab or the company before you go in. Maybe there's a press report you can read. If they have public tools or data sets available, look at them so you can reference them naturally in conversation. It shows really genuine interest and that you've done your homework, which I think puts you ahead of most candidates. All right, so common pitfalls and our pet peeves. Let's talk about that. I think, there's many mistakes you can maybe make in an interview. You might over rely on pipelines without understanding the individual steps. That's a big one. Again, you want to show that you understand the why behind the pipeline. Why is it structured the way it is? Not just that you can run it. And then another thing you can talk about is computational resources. Show them that you know about scalability, optimization, memory usage when relevant. You know that you can make it work, but you also want to show them that you can make it work efficiently.
Exactly. And of course, forgetting the biology is another huge one. Every analysis should answer a biological question, not just produce numbers. Another one is when candidates talk at length without pausing to check if they are answering the actual question being asked. So it's okay to stop and say, is this what you're looking for? Rather than going on for 5 minutes without stopping.
Yeah, for sure. And then here's another pitfall. Candidates who can't explain their own thesis or main project work clearly and concisely. So if you spend years on something, you should be able to give a compelling 2-minute overview. When someone can't do that, it makes me wonder how well they actually understood the work that they spend quite a bit of time on, right? So in advance, you want to practice your elevator pitch, make sure that's solid.
Yeah, practice everything. And last thing, not asking any question at the end. This is supposed to be a conversation. You really need to know if this is a good fit for you as well. So come with genuine curiosity about the team, the work, and the environment.
Yeah, for sure. They are also trying to impress you, right? It's not just you trying to impress them. Ideally, they need to impress you as well. So for final thoughts, I want to say, they expect you to be junior or intermediate level. So you don't need to try and fake expert knowledge. You want to show enthusiasm for learning because this field moves so fast. You always need to learn and upskill. That's just expected from you. And be honest about your experience. I think depth in one area is better than shallow knowledge everywhere.
And the secret to all this, well, there is no secret, to be honest. But the best you can do is to demonstrate that you can learn, communicate, and think critically about data. Technical skill can be tough. Biology and bioinformatics tools change every 2 years. Problem solving
Right.
mindset cannot. So don't forget to follow up as well with a thank you email that references specific things that you have discussed. That is nice because it seems that you really think about this position and that the small touch can make a difference.
Right. I totally agree with that. Good luck out there, everyone. Let us know how it's going.
Yeah, you got this. Thank you for watching.
Thank you for watching.